Over the past few years, frontline clinicians have reported a disturbing pattern. Working-age adults, typically at low risk for severe malignancies, are suddenly presenting with aggressive, late-stage tumors that spread at an unnatural speed. It's a phenomenon that has colloquially become known as turbo-cancer. This clinical observation didn't emerge in a vacuum. An independent reanalysis of the mRNA clinical trials revealed an absolute risk increase for serious adverse events. For every 800 vaccinated individuals, there is one excess serious adverse event, a rate that forces a closer look at the interaction between the platform and the body. This risk fueled explosive internet theories. The most prominent claim is that mass-produced vaccine vials are contaminated with residual bacterial DNA, specifically SV40 plasmids, that purportedly slip into human cells, mutate the genome, and directly spawn these aggressive tumors. While the anxiety surrounding a 1 in 800 complication rate is based on clinical trial data, accepting internet hyperbole without verification obscures the truth. To understand what is happening inside these patients, we have to look past the panic and examine the actual, provable biology. This graph tracks residual DNA concentrations across various vaccine batches. The strict international safety limit is set at 10 nanograms. Independent deep sequencing analysis proves that the residual DNA in these vials is highly fragmented and falls well below this safety line. These fragments are too degraded to enter a nucleus and mutate our genome. But if the DNA isn't mutating cells, what about the epidemiological studies claiming a 27% spike in cancer incidence following vaccination? That statistic comes from enormous healthcare databases in South Korea and Italy. When independent statisticians audited that data, they found a severe mathematical error. This chart compares historically expected cancer rates with the control group used in the Asian studies. The unvaccinated control group is missing nearly half of its expected baseline. The study compared vaccinated patients against an impossibly healthy control group, inflating the relative risk. The perceived spike in short-term cancer is actually driven by medical surveillance bias. Highly vaccinated populations utilize healthcare screening more frequently. They get more mammograms and colonoscopies, leading doctors to detect pre-existing subclinical tumors earlier than they would in people who avoid the doctor. If the population statistics are a mathematical illusion and trace DNA isn't altering our genome, we have to look elsewhere. The aggressive tumor progressions observed by oncologists aren't being born from a new genetic mutation. They are being accelerated by a microscopic shift in the human immune system. The mRNA lipid nanoparticle platform does not spontaneously turn healthy tissue into cancer. Instead, it is a highly specific immune modulator. It alters the body's internal microscopic ecosystem, and that ecosystem is what keeps dormant disease in check. To understand how, we have to look at a protein called p53, known as the guardian of the genome. If a cell sustains catastrophic damage, p53 halts the cell cycle and forces the cell to self-destruct, a process called apoptosis, before it can multiply. Independent molecular biology research reveals a disruption to this security system. The host-manufactured synthetic spike protein contains a specific segment called the S2 subunit. This subunit physically wedges into the cell's signaling architecture, blocking the interaction between p53 and its regulatory nodes. It functionally paralyzes the cell's ability to transmit the halt signal. The clinical consequences of this localized suppression are severe. When scientists expose laboratory cancer cells to DNA-damaging chemotherapy, the cells don't die. Because the p53 alarm is muted, existing cancer cells gain a survival advantage, replicating through what should be a lethal chemical attack. The intervention doesn't create the cancer from scratch. By blinding the p53 security system, it simply cuts the biological brakes on cells that were already heading toward malignancy. While the cellular alarms are muted locally, a shift occurs across the systemic immune system. Traditionally, the body neutralizes viral or mutated threats using aggressive inflammatory antibodies known as IgG1. These IgG1 antibodies act as the body's cavalry, aggressively latching onto targets and recruiting killer cells to dismantle the threat. However, longitudinal cohort studies document an unusual immunological response following repeated doses of mRNA boosters. Over time, the immune system undergoes a shift. skewing its antibody production away from the aggressive IgG1 and toward a subclass called IgG4. IgG4 is biologically unique. It is a non-inflammatory, tolerogenic antibody, essentially a biological white flag. The body normally uses it to build tolerance to harmless environmental allergens, but here, these passive IgG4 shapes occupy the surface binding sites of a tumor cell, blocking the aggressive IgG1 cavalry from attaching and doing their job. Combine the localized blinding of the p53 cellular security system with the systemic exhaustion of the immune cavalry, and you get a devastating biological environment. Pre-existing dormant tumors are granted an immunological free pass, providing the conditions needed for the rapid growth clinicians are reporting. The scientific consensus requires precision. The mRNA platform acts as an accelerant for existing neoplastic disease, rather than a creator of new tumors. This leads to a biological paradox. The same immunomodulatory power of the mRNA platform has demonstrated the capacity to improve survival for certain advanced cancer patients. In a clinical trial, end-stage melanoma and lung cancer patients were given standard COVID-19 mRNA vaccines alongside targeted immune checkpoint inhibitor therapies. The survival times for these patients were significantly extended. The lipid nanoparticles within the vaccine trigger a surge of chemical signals that effectively resets the cancer immunity cycle. This primes the body's T cells, causing them to swarm and dismantle heavily shielded tumors that the immune system previously couldn't see. An intervention powerful enough to orchestrate a body-wide immune response to destroy terminal cancer is, by definition, an active biological agent with systemic effects. We have to contrast that targeted oncological power against the broader population data. We are looking at a 1 in 800 serious adverse event rate, alongside sustained global excess mortality across working-age populations that institutional bodies are currently investigating. Yet, objective, ongoing safety evaluations are hindered. Independent medical professionals point out that institutional structures and funding models have made it difficult to conduct an open pharmacovigilance analysis of the mRNA platform. The biological risks identified, from p53 suppression to IgG4 tolerance, shift the risk-benefit calculation for the platform. These mechanisms suggest that the indiscriminate use of this technology may carry unintended consequences for long-term health. The future of mRNA technologies may instead lie in targeted precision oncology. In those terminal cases, the capacity to drastically alter the human immune system is a specific therapeutic objective, rather than an unpredictable risk to a health